This is the second lecture recording for the module with bacteria and viruses. And for this recording, I'm going to focus only on viruses. So I'm going to discuss the basic structure of a virus, the viral reproductive cycle as it applies to bacteriophages, which are the viruses that affect bacteria. And then I will talk about HIV, influenza, and SARS, which causes COVID-19. So first of all, we talked about this at the beginning of the semester, but viruses are not considered living organisms because they do not reproduce on their own. Now, they do possess some of the characteristics of life, such as the ability to mutate, but they need the proteins and cellular machinery in the host cell in order to make more viral genetic information and viral proteins. Therefore, they are not considered living. Now, there are several components to the structure of a virus. So first of all, we have viral genetic information, and that can take the form of DNA or RNA. So I'm just going to write the viral genome. So it can be single or double-stranded DNA or RNA. So it just depends on what type of virus you are talking about. Now, another component of a virus is the capsid, which is the protein sheath that is covering the genetic information. So you can kind of think of it like a cell wall, but it is not the same structure as a cell wall. So we will say it is the protein sheath enclosing the viral genetic information and enzymes. Now, not all viruses have a huge number of enzymes. Only a few viruses carry their own enzymes. So we have the viral genome, the capsid. Now, some viruses have what we call an envelope. So you'll see that in HIV in my picture here. Now, the viral envelope has, different structures depending on which virus you're talking about. But a lot of times it's like a sugar lipid combination or a sugar protein combination. And they usually have, it says envelope protein. They usually have proteins on them that help them attach to the host cell. So I'm going to write some viruses include a viral envelope. Okay, now remember, this is not the same as a cell wall, but it's similar. Or actually, I guess you could consider it kind of like a membrane. Now, there are different groups of viruses, and within each group, we have a variety of different viruses that are included, right? So first of all, we have HIV. So that's kind of a virus on its own. So HIV, if you notice in the picture here, has RNA as its genetic information. It also contains an enzyme we call reverse transcriptase. It has a lipid membrane, that's like the viral envelope, with glycoproteins extending off of the lipid membrane. And those are to help HIV attach to the host cell. You'll see it also has a capsid. Now, a bacteriophage, we also call that just a phage, and that infects bacteria. So when I talk about the life cycle of bacteriophages, I will mostly talk about the kind that infect E. coli, because that is where we have done a lot of research on viruses. We have the influenza virus, obviously causes the flu, but there's actually 4 different types of influenza virus that we now recognize. There's influenza A, B, C, and D. Now, not all of these types infect humans. However, there is the potential for, I believe it's influenza D that does not currently infect humans, but there's always a chance that it will. And we'll talk about that when we get to influenza. But it has RNA, it also has a capsid, glycoproteins on the outside. We have adenoviruses, which cause like the common cold. So a lot of people take antibiotics for colds, but most colds are caused by a virus. So antibiotics don't help you fight off the cold. And those have DNA. And again, so what you're seeing here, so these do not have an envelope on them. Some of them do, but a lot of them do not. They have the DNA, the capsid, which is protein, and glycoproteins coming off of those. Now, notice that they all have glycoproteins. Those are used in cell signaling or attaching to the host. Now, there's another term here, capsimer. So capsids are made of subunits, like all these little units put together, and those are what we call capsimers. We have the tobacco mosaic virus, which is the first virus that was ever discovered. So obviously it infects tobacco. There are forms of it that infect other plants, and they can actually be very detrimental to agriculture. And then finally, Ebola, which you've probably heard of, but it hasn't been an issue. It's been a few years now. I think COVID kind of took over that spot. So here we are looking at the viral reproductive cycle. Now keep in mind that I am talking about bacteriophages. Okay, this is the viral reproductive cycle for bacteriophages or phages. And again, these are the viruses that affect bacteria like E. coli. So there are 2 different phases in this reproductive cycle. There's what we call the lytic cycle and the lysogenic cycle. So we will talk about the lytic cycle first. Now, first of all, when a virus comes up to a host cell, now keep in mind that specific viruses infect specific host cells, or organisms, I should say, but not just a specific organism, but certain types of cells within that organism. Like for instance, the SARS virus affects cells that are in like your upper respiratory tract. So the first step in any viral reproductive cycle is attachment. So the virus has to attach to the host cell. And this is usually done with glycoproteins. So in my picture here we have the phage. Remember, bacteriophages are often called just phages. And it is attaching to a bacterial cell, E. coli. So it attaches to the host cell, then it injects its genetic information, which in this case is DNA. Okay, so we have attachment. And then I'm going to put here, virus injects its DNA. Into the host cell. Okay, so it attaches, the virus injects its DNA. So now we're down to this middle area here. Now, what happens is that in the case of a phage, it also injected some enzymes that break up the bacterial DNA. So we're going to write viral enzymes break up the bacterial DNA, okay? So at the bottom, it says new phage DNA and proteins are synthesized and assembled into phages. So basically what that means is the virus has taken over the bacterial cell and it needs the enzymes inside that bacterial host in order to make more viral genetic information and proteins. So I'm going to write virus uses the bacterial enzymes Okay, so the virus uses bacterial enzymes to make more viral DNA and viral proteins. The next thing that happens is that viral DNA and proteins are packaged into new viral particles that we call virions. A virion just basically means a single virus particle. So viral DNA and proteins are packaged to new viral particles or virions. And then finally, the cell lyses or bursts. This is the bacterial cell, releasing all of those new virus particles. Okay, so this is the lytic cycle. The bacterial cell is infected by the phage virus. The virus injects its DNA. The viral DNA breaks up the bacterial chromosome. The virus uses the bacterial enzymes and machinery to make more viral DNA and proteins. These are packaged up into new viral virus particles or virions. The bacterial cell lyses or bursts, and those virus particles are released. Now, there is another part to the viral reproductive cycle of a bacteriophage, and this is called the lysogenic cycle. Now, the thing to remember about the lysogenic cycle is it does not result in the death of the bacterial cell. Okay, the bacterial cell does not lyse. So it begins the same way. The phage attaches to the bacterial cell and injects its DNA. Okay, so let's look at this middle area right here. Okay, I'm looking right here. Now, it says certain factors determine whether the lytic cycle is induced or the lysogenic cycle is entered, meaning that once a bacterial cell is infected with this phage DNA, it can either enter the lytic cycle, where it will burst, or the lysogenic cycle that it can remain in for years. And it just depends on environmental cues, like say if there's stress in the environment, right? Maybe the bacterial cells are stressed, they don't have enough nutrients or something like that, then the virus will enter the lytic cycle. But if the bacteria is in like a good environment, then the virus will probably enter the lysogenic cycle. It also depends on what specific type of bacterial virus we're talking about. Okay, so for the lysogenic cycle, the first several steps are the same as the lytic. The phage attaches, it injects its DNA, but then with the lysogenic cycle, the viral DNA does not break up the bacterial DNA. Instead, the viral DNA inserts into the bacterial DNA. So I'm going to write that here for you. So I'm going to start at the beginning. So phage attaches to bacterial host. Viral DNA is injected. Okay, but if the lysogenic cycle is entered, then the viral DNA integrates or combines into the bacterial DNA. Now, this viral DNA is called a prophage when it's in the lysogenic cycle. Now, every time the bacterial cell reproduces, it makes more viral DNA. Remember, bacteria reproduce asexually. It's called binary fission. So a bacterial cell will duplicate its genome, its DNA, make another circular chromosome, and then that bacterial cell will divide in half. That's just a simplified explanation. But what you need to know here is that every time a bacterial cell duplicates its DNA and divides, it is also duplicating that prophage. So it's making more bacteria with the viral DNA in it. So every time the I know I just wrote a lot here, so you'll probably have to pause this video. Okay, so the bacterial cell, it's reproducing, it's making more bacterial cells. They're like clones of the original because this is asexual reproduction. Now, all of those new bacterial cells also have that viral DNA, okay? Now, if you look at the top here, it says occasionally a prophage exits the bacterial chromosome, initiating a lytic cycle. So you go down to this middle picture here. Now remember, the viral DNA is that blue circle. So it comes out of the bacterial chromosome and it starts the lytic cycle where it will then fragment the bacterial DNA and kill the bacterial cell. So a prophage okay. So I want you to know the basic steps of the lytic cycle and the lysogenic cycle. And I want you to remember that this occurs with bacteriophages. So now we are going to look at HIV. So HIV, human immunodeficiency virus, actually started in apes in Africa, and in primates, it's actually in monkeys as well. In primates, it's called SIV, simian immunodeficiency virus. And what happened is a long time ago, I can't remember when it was, like mid 1900s, because humans were consuming primates, the SIV entered the human body and mutated to a form that is easy to spread between humans. So that is how we got HIV. Now we call HIV a retrovirus. First of all, its genetic material is RNA. And we call it a retrovirus because it contains an enzyme called reverse transcriptase. Now, normally, when proteins are made, a cell takes genetic information in DNA, it takes that genetic information, produces an RNA. Remember, those are both nucleic acids. And then the genetic information from RNA is used to make proteins. Well, with HIV and other retroviruses, this process is reversed. The HIV will inject RNA, but it needs DNA to be able to enter the nucleus of the host cell and have its genetic information copied. So with HIV, it goes from RNA to DNA. So the opposite of what we normally see in cells. So viral DNA is made from viral RNA. The DNA of the virus enters the nucleus of the host cell and inserts into host cell DNA. Now, what that means is when the host cell takes that genetic information and uses it to make proteins, it will take the viral DNA and make viral proteins. So let's go to this image here. So first of all, as with all viruses, attachment is the first step. Now, HIV is specific to human hosts. It doesn't attack just any cell in the human body, it attacks cells in the immune system. So T cells and things like that. So we have our HIV virus, it has RNA, it attaches to the host cell because the receptor or glycoprotein on the HIV virus matches the receptor on the host cell. It injects its RNA. So actually with HIV, I believe the whole virus enters, but regardless, the RNA becomes exposed. Also, the viral enzyme called reverse transcriptase. Okay. Now, reverse transcriptase makes viral DNA from RNA. Now, it's usually the opposite. The viral DNA enters the nucleus of the host cell and it incorporates into the host cell DNA, just like we see with a prophage. Now, when the host cell starts duplicating its own DNA and making RNA for proteins, it also duplicates that viral DNA and makes viral RNA for proteins. So you have viral DNA, from that you get viral RNA, that leaves the nucleus, and the viral RNA is used to make viral proteins. Now remember, surrounding the genetic information of a virus is the capsid or protein sheath. So that is what a lot of these proteins are being used for. So now you have the proteins, those are the bluish circles here. You have viral RNA, they get all packaged up in a capsid, and they still have reverse transcriptase, also a protein, right? Because it's an enzyme. And then what happens is what they're calling release in this picture. So the new HIV viral particle leaves the host cell. Now, it actually damages the host cell enough that it no longer works. It's not necessarily causing lysing at this point, but it has damaged the cell in the immune system to such a point that it no longer functions correctly. Okay, so now we are going to look at influenza, which is also, an RNA virus. Now, influenza is not a retrovirus, right? It contains RNA, but it doesn't have that reverse transcriptase enzyme. So it enters an epithelial cell in the upper respiratory tract. So a lot of times people say they have the flu when they have vomiting and nausea, but that's actually not the same as influenza. Influenza is a respiratory illness. So it says the virus enters an epithelial cell. So in this case, the virus is not injecting the RNA, the whole virus particle is entering the epithelial cell. So that's what you see here for number one. The virus particle approaches the host cell. So the virus enters the host epithelial cell. Now, once it enters the host cell, the viral RNA and what we call RNA polymerase are released into the host cell and enter the nucleus. Now, RNA polymerase is used to make RNA from DNA. So actually, it's used to make messenger RNA, excuse me. So once the viral RNA is into the host cell nucleus, that RNA polymerase makes the next type of RNA on the way to producing proteins. Now you have more viral RNA, more viral proteins. They get packaged up into a new viral particle. Now, the virus particle leaves the host cell, but it is enclosed in the host cell plasma membrane. So we call this budding. Okay, so it's almost like it's cloaked in the host cell plasma membrane. So just to review what I just went through with influenza, the influenza virus enters the host cell. The viral capsid and the envelope, they're degraded, and the viral RNA and RNA polymerase are released. These enter the host cell nucleus. They are used to make more viral RNA and viral proteins. These are packaged up into new virus particles, and then they bud out of the host cell. And they're encapsulated in the host cell plasma membrane. Now, the last virus we will talk about is the SARS virus, which is what causes COVID-19. Now, there are many types of SARS viruses, most of which infect mammals. Now, with COVID-19, we actually are pretty confident now that it was passed from bats to humans. And I'm not saying that people were necessarily eating bats, but they were in close contact with them. So SARS attacks cells in the lower respiratory tract, but we've also found that it can enter cells in the upper esophagus, the digestive system, heart, and kidney. That is why you see so many different symptoms of COVID-19. And in addition to that, the SARS virus is mutating, or the COVID-19 virus is constantly mutating, and that is why we see different symptoms as well. Now, this is a complicated picture, but there are just a few things I need you to know about this. So first of all, just as with influenza, the entire SARS particle enters the host cell. So I'm going to write that here for you. Okay, so it enters the host cell intact. So then the viral capsid is degraded and the genetic material and enzymes are released. Okay, that's what you're seeing in step 2 here. Now, the only thing you need to remember here is once the genetic material and enzymes are released, additional viral genetic material and proteins are produced. New viral particles are assembled. Whoops. And then the virus is released through exocytosis, which you should know what that is. If not, you can go back and review the information from cell structure. Okay. So just an overview of the entire virus presentation. You need to understand the basic components of a virus, meaning the capsid, which is made of proteins, the genetic material, which can be DNA or RNA, and the occurrence of an envelope in some viruses, and then glycoproteins that help them recognize the host cell. Remember that viruses are specific to certain organisms and certain cells within the organisms. For instance, HIV affects cells in the immune system. SARS and influenza affect epithelial cells in the upper and lower respiratory tracts. Okay, you need to know the lytic and lysogenic cycles for a bacteriophage. Lytic causes lysing of the bacterial cell or bursting. Lysogenic can be maintained for years. It's kind of like a dormant state. And then you need to know what an HIV virus does, the fact that it is a retrovirus, and then the basics of influenza and SARS infections. And that is it for the virus presentation.